Hello students, how are you all doing? I hope all of you are fine. Students, this is my 6th class on the chapter Sexual Reproduction in Flowering Plants. In this class, I am going to discuss megasporogenesis and then development of embryo sac. So, in your theory examination, they will ask you questions like what is megasporogenesis, 1 marks question. Explain megasporogenesis with a neat labeled diagram, 3 marks question. And also they will ask you to write a neat labeled diagram of embryo sac, which already I have explained that in your previous class, in my previous class. So, in order to make you understand the process of megasporogenesis, I would like to compare the process of megasporogenesis with that of the microsporogenesis. Already we have discussed the process of microsporogenesis and you have understood that well. So, in order to give you comparative idea, so that you will also be able to write the differences between microsporogenesis and megasporogenesis, a very simple difference. And at the same time, you shall have a deeper idea, deeper level of understanding regarding to the differences between the microsporogenesis and megasporogenesis. For that reason, I have written the process of microsporogenesis here. Okay, here there is a microspore. Let me begin here. So, here there is a section of anther where pollen grains or microspores are not yet formed. So, these are 4 microsporangia, you know that. Each microsporangia has got a sporogenous tissue. I have taken one cell from this sporogenous tissue. This one cell will undergo meiosis, first meiotic division. It has given rise to 2 cells, dyad of microspore, and further it will undergo second meiotic division. So, it has resulted in a tetrad, microspore tetrad. You have studied this. And as anther dehydrates, the microspores separates, and then out of these 4 microspores which are haploid, each one of them, they are all haploid microspores. They are the result of meiotic division of one of the cell from the sporogenous tissue, and all the cells in the sporogenous tissue will undergo the process of meiosis, and thousands of microspores are formed in each of the microsporangia, which is also referred as a pollen sac. So, I have just taken one cell here, and one cell has given rise to 4 microspores, and then I have considered one microspore where this haploid microspore will undergo mitotic division to give rise to one larger cell, which is referred as a vegetative cell, and one smaller cell which is referred as a generative cell. The generative cell, smaller one, is a spindle shaped, and actually it floats in the cytoplasm of the vegetative cell. It is very large. And then I have explained you that this smaller generative cell will further divide by the process of mitosis to give rise to 2 male gametes, and meanwhile there is a formation of the exine, sporopollenin layer, the exine of the pollen grain, and finally the pollen grain is being formed. And even I have told you that in more than 60% of the angiosperms, pollen grains are released in a 2-celled stage. Okay, here too already sporopollenin layer will have been formed like this, and it will be released in the 2-cell stage, and whereas in remaining 40% of the angiosperms, pollen grains are released in a 3-celled stage where vegetative cell and 2 male gametes are there. And you are going to understand the role of vegetative cell when I do explain the process of fertilization in case of the angiosperm. So, this is the brief idea of the microsporogenesis, the formation of microspores, and the development of one of the microspore to a male gametophyte, that is the pollen grain. So, I will compare this process of microsporogenesis and the development of the microspore into a male gametophyte, the pollen grain, to the megasporogenesis. Okay, I will explain the way how the megasporogenesis take place, and then how megaspore develop into the female gametophyte, which is also referred as a embryo sac. Already you are all familiar with the structure of embryo sac, and I shall give you the way how the development of the embryo sac will take place. Okay, and you are also familiar regarding to the structure of ovule. Actually, you have studied the structure of anatropous ovule, and in order to make you understand the structure of anatropous ovule, I have explained you the structure of this type of ovule, the orthotropous ovule. And here this is the ovule where embryo sac is not yet developed. You know, there is a presence of the integuments, a funicle, chalaza. Here the tissue which I have written by using blue color chalk paste, this is nucellus, right? So you cannot compare this nucellus to this sporogenous tissue. Here in case of a microsporangium, all the cells of this sporogenous tissue will enter the process of meiosis. And that results in the formation of thousands of microspores, pollen grains, in each one of the microsporangium in the anther. So here anther has got 4 microsporangium, whereas here there is a presence of in ovule there is a presence of one tissue, that is the nucellus. Only one nucellus tissue is there, one unit is there. So this nucellus tissue or the cells present in the nucellus are involved in providing the nourishment for developing megaspore. One of the cell in the nuclear tissue will differentiate. I will take out that cell. Okay, one of the cell in the nuclear tissue, this is the nucellus. Let me consider a cell somewhere here, a cell towards the micropylar end. Towards the micropylar end, this is the micropyle. You know that the cell towards the micropylar end will enlarge. It is having a dense cytoplasm with reserve food material. It is having a larger nucleus, and this cell, a single cell, will differentiate, and this is the cell which is going to enter the process of meiosis for the formation of one single megaspore. Okay, this is a main difference. Now, it is not given in your textbook. You have to understand this. The entire sporogenous tissue, all the cells will enter the process of meiosis to give rise to pollen grains. Here, the nucellus, a tissue which is involved in providing the nourishment for the megaspore, only one of the cell will enter the process of meiosis, and we call this as a megaspore mother cell. Simply, I will write the abbreviation like megaspore mother cell, but you have to write full form in your examination. It is a megaspore mother cell. Whereas here the tapetum, the layer which I have marked by using yellow color chalk piece, we call that as a tapetum. This tapetal layer surrounds the sporogenous tissue, and the tapetum is something which is going to provide the nourishment for the sporogenous tissue. Here the nuclear tissue is going to provide the nourishment, and one of the cell of the nucellus towards the micropylar end will enlarge, will have more reserve food material with the dense cytoplasm and with a large nucleus, and this is the one single cell which is going to enter the process of meiosis, and it is megaspore mother cell. You make out the difference, try to understand the difference. So this megaspore mother cell, as I have told you, will undergo meiosis, first meiotic division. So in your textbook what they do, this is the megaspore mother cell. Let me give you the information how it is given in your textbook. Okay, so surrounding this megaspore mother cell, there is a presence of the other nuclear cells like this. They have given like this. These are the other nuclear cells. So one of the cell is to be taken as a megaspore mother cell. Okay, they have given the diagram somewhat like this. Right. So this is megaspore mother cell. Megaspore mother cell. So this megaspore mother cell, what happens? As I have told you, it will enter the process of meiosis. There occurs first meiotic division, and due to this, 2 haploid cells are formed. Okay, megaspore dyads are formed, and again you have to show the nuclear cells. You have to represent the nuclear cells in this way. Okay, these are the nuclear cells like this. Okay, so this is megaspore diode. Two cells are there, and then what happens next? This megaspore diode, both of these cells, haploid cells, will enter the second meiotic division like this, so that the cells are arranged one below the other like this. 4 megaspores are formed, and again you have to represent the nuclear cells surrounding these 4 megaspores like this, right? You have to write this diagram in your examination, and the diagram may be given as I have been telling you in each of my class. Like the diagram will be chosen directly from the textbook, and there may be a markings like a B, C, D. You will have to identify the parts. So this is a megaspore tetrad. 4 cells are there. Megaspore tetrad diode. I write here. It's a megaspore tetrad. Megaspore tetrad, and now 4 megaspore cells are formed. Out of these 4 megaspore cells, 3 of them will degenerate, and you shall ask me the question, why these 3 of them degenerates? So you see here, students, one ovule will have a level of nourishment. A ovule can support the development of one seed, a development of one embryo. So if many embryos are formed in one ovule, what may happen? The food reserve which is present in the nucellus. Nucellus is the tissue which is going to provide the nourishment for the developing embryo after the fertilization, pollination and fertilization. So if all these cells, more than 2, 3, 4, 5, many cells gives rise to many megaspores, and all the megaspores will undergo the development to give rise to male gametes, and if many embryos are formed, what may happen? The nucellus tissue which is there in one ovule may not be able to provide the nourishment for all the embryos. That's not possible. So a ovule is completely capable of giving rise to a single seed, a single embryo. So you see here, nature is determining that only one of the megaspores formed out of 4 haploid megaspores, only one of the megaspore will enlarge, and remaining 3 will disintegrate. They will wither away. They degenerate. And this one megaspore haploid will develop to give rise to embryo sac. Okay, I consider this one megaspore like this. Megaspore. So this one megaspore has to give rise to the embryo sac. So what have we understood until now? We have understood the process of megasporogenesis. And I will give you the information the way how it is given in your textbook. What is megasporogenesis? Megasporogenesis is the process of formation of a megaspore from megaspore mother cell. 1 marks question. What is megasporogenesis? Megasporogenesis is the process of formation of a megaspore, a single megaspore from megaspore mother cell. Okay, and this megaspore mother cell, at the time of formation of the megaspore mother cell, 3 megaspores degenerate and only 1 megaspore give rise to a embryo sac. This type of development of the embryo sac, we call that as a monosporic development. Monosporic. What is monosporic development of the embryo sac? Monosporic development of the embryo sac is a phenomenon where one of the megaspore gives rise to embryo sac, while 3 other megaspores formed by the process of meiosis undergo disintegration. Okay, they may ask you 1 mark question, what is monosporic development of the embryo sac? So like that. So now we have got the definition for megasporogenesis, and now the explanation continues. One of the cell towards the micropilar end, I write it by using the yellow color chalk piece. One of the cell toward the micropilar end you write down the information the way how I explained. I have given you the concept. I am giving you the sentences which are there in my notes. One of the cell towards the micropylar end will give rise to megaspore mother cell. Megaspore mother cell is larger with the dense cytoplasm and larger nucleus. This will undergo first meiotic division to give rise to the megaspore dyad, and it enters second meiotic division. This is first meiosis and this is second meiosis. Second meiotic division to give rise to megaspore tetrads, and then 3 megaspores degenerate and only one give rise to a embryo sac. This is a monosporic method of development of embryo sac, and now this haploid megaspore has to give rise to the embryo sac or a female gametophyte. So the process of development of female gametophyte or embryo sac from megaspore, we call that, we have to refer that under the heading development of embryo sac. So now let me explain you how the development of embryo sac take place. Students, you have to remember this diagram from here to here. So these diagrams are there in your textbook. This one is not there. Again, this diagram is there. Okay, you will have to practice to write this diagram. So now let me consider this one megaspore, and you have got clear idea how this megaspore is being formed. This is haploid structure. Haploid is represented by small n. You know that. What is this? Megaspore. Megaspore. So what happens? This megaspore, it is still there inside somewhere here. It is there inside. Let me write it by using the yellow color chalk piece. It will be much conspicuous so that it will be easy for you to refer. So you see here, somewhere here the megaspore is there. So now what happens? This megaspore will start to divide by the process of mitosis. So you know what is mitosis? Mitosis is the process where a cell gives rise to 2 daughter cells, right? 2 daughter cells like this. So at the time of division of a daughter cells, I will write it here, 2 daughter cells are formed where the equal number of chromosomes are distributed. So at the time of division, what happens? First, there occurs the nuclear division, what we also call as a karyokinesis, and then there occurs a cytoplasmic division, what we call as a cytokinesis. Karyokinesis and cytokinesis, you have studied that in your first year PUC. Here what happens? At the time of the development of the megaspore to give rise to embryo sac, only karyokinesis, nuclear division will take place, and the nuclear division is not followed by the cytoplasmic division, that is the cytokinesis. So this type of mitotic division here, we call that as a free nuclear division. What is it? It's a free nuclear division. So this embryo sac will undergo free nuclear division so that the 2 nuclei are formed. It's a mitotic division. It's mitosis, not meiosis. Mitosis. Meiosis take place in microspore mother cell, right? Sorry. Meiosis take place in microspore mother cell. Here, in my explanation, I am explaining the process of megasporogenesis. Meiosis take place in megaspore mother cell, even in microspore mother cell. So meiosis will take place in megaspore mother cell, but not in megaspore. Megaspore undergoes ordinary mitotic division of a free nuclear kind to give rise to embryo sac, which is the type of division which take place at the time of development of embryo sac. Usually you will get confused and write it is meiosis in your NEET examination. No, at the time of development of embryo sac, megaspore divides mitotically by free nuclear division. When meiosis takes place in megaspore mother cell, so as to give rise to 4 megaspores, wherein only 1 will retain itself and 3 are going to degenerate. So that's why I have written all this diagram to give you comparative idea, to give you a whole concept as a package so that you can compare and you can think. Okay, so here it's undergoing mitosis, free nuclear division, 2 nuclei are formed. So next again, further these 2 nuclei will again divide so as to form 4 nuclei. This will further divide by the free nuclear division so as to give rise to, all the nuclei will divide so as to give rise to 8 nuclei. How many nuclei are there? Totally 8 number of nuclei are there. All these nuclei are the result of free nuclear division. Later on what happens? Except 2 cells, for all the other cells, there begins a formation of cell wall. Cytoplasm surrounds each of the nucleus and there begins a formation of a cell wall. And now what happens? One of the cell will develop into egg cell and 2 of them develops into synergids like this. So here it is given in lower side, I am giving the orientation towards upper side, then we have to invert that, you have to consider this way. So here this embryo sac is with respect to anatropous ovule, where the ovule is bent like this. Here I am explaining with respect to orthotropous ovule. Even in your textbook the explanation is like this. But finally when they write the structure of embryo sac, they consider the diagram like this. You shall not confuse. Here it is towards upper side, here there is towards lower side. Why? Because here they have considered the embryo sac in anatropous ovule, but the explanation of the development of the embryo sac is with respect to orthotropous ovule. Okay? Orthotropous ovule over the micropyle is toward the upper side. So how many cells are over? 3 cells have been utilized. And remaining 3 cells are utilized to form this one, egg and synergid, which is also referred as egg apparatus. Okay, and these 3 cells, what may happen? Simply I am marking like that. They come to the opposite pole, they segregate towards the opposite pole, and they are going to form the antipodal cells. Right? And remaining these 2 cells, here cytoplasm is not, the cell wall is being not, cell wall is not being laid out. Cell wall formation has not taken place with respect to these 2 nuclei. These two nuclei with the cytoplasm will come together and there occurs a fusion of the cytoplasm so as to make one composite cell. One composite cell with 2 nuclei, we call this as a central cell. Okay, and you consider this in a twisted way. So you see here, these are the 2 synergids. Okay, let me mark it by using the same color chalk piece. These are the 2 synergids like this, and this is the ova like this. So here you take it in an inverted way, as in case of anatropous ovule. These are the 2 synergids. This is the egg cell, ova egg cell, and these are the central cell. Okay, central cell, and here these are the antipodal cell. So the development of the embryo sac take place in this way, right? By the process of free nuclear mitotic division. And now when we come to the structure of the embryo sac, synergids helps in the process of fertilization by guiding the male gametes to the ova. That's how the sentence is being given in your textbook as well in my notes. You will understand that when I discuss fertilization, synergids take up the role of guiding the male gamete to the ova, and synergids are also involved in protection of ova. Okay, and functional significance of the antipodal cells is not being understood, and I will tell you what is the importance of the central cell. Okay, when I do discuss the fertilization, process of fertilization and the development of the ovule into the seed, and the development of egg into the embryo, fertilized egg, after the fertilization the egg cell will become zygote, it will develop into the embryo. While I explain the development of the embryo and the transformation of the ovule into the seed, I will explain what is the function or a functional significance of the central cell as well the synergids. For time being, as per the requirement of this class, what all have you understood? You know to write and need to label the diagram of the embryo sac and explain its structure. You know to explain the development of embryo sac, or it is also female gametophyte. I have told you why it is referred as a female gametophyte. It is a haploid phase of a life cycle of a plant, a small structure which is giving rise to a female gamete. So, it is a female gametophyte. So, the development of the female gametophyte from the megaspore, or development of embryo sac from the megaspore, and in addition to that you have understood the megasporogenesis, right. So, now let us try to understand the difference between microsporogenesis and megasporogenesis as per the content of your textbook and the information given in your notes. Microsporogenesis occurs in microsporangia, which is present in anther. Megasporogenesis occurs in the ovule. Microsporogenesis gives rise to 4 microspores, where each one of the microspore gives rise to a pollen grain. During megasporogenesis, one of the megaspore develops into embryo sac. So, that is the plain information which is available for you. But in addition to that, let us try to understand, let us try to have an idea much more differences. You can make much more differences, like already I have told you that this only one here in anther 4 microsporangias may be there, but actually here there is a presence of, in the ovule, only one megaspore development can take place, because the nuclear tissue will have a capability of providing the nourishment for one embryo so as to sustain one seed. But whereas here microspores, they are not involved in a development of the embryo, they simply provide the male gametes for the ovule to the embryo sac, where the embryo development will take place. Okay. So, you please read your textbook after listening to me. Go through my notes. You will have a thorough idea and understanding. Okay, students, in my next class, I am going to continue the discussion of the aspects of sexual reproduction in flowering plants, and I hope you have all understood what I have taught you in this class. Please refer your notes, textbook. Thank you all for listening to me. Thank you.